Hello everyone. My name is Veera Baladandayuthapani. I'm a professor in the Department of Biostatistics, and I welcome you all wherever you are in the cyberspace watching this video. And I'm glad to be talking to you, even though it's virtually. I'm glad to be talking to you. So before I start my spiel, I just wanted to know a little bit about me. So I got my PhD a couple of generations ago now from Texas A&M, and my dissertation was in Bayesian bioinformatics. I started here around a couple of years ago, and I used to work at MD Anderson Cancer Center, which was one of the premier cancer centers in the world. It still is, and I was there on the faculty for 13 years, and that's where I really saw how biostatistics and biostatisticians can make a real impact. So my own research focus is in big data integration. I'm a Bayesian statistician with the flavors of machine learning networks, with primary applications being genomics and imaging, and my sort of the disease area that I sort of focus on is in cancer. I see myself as a passionate collaborative data scientist, and most of the methods, or most of my methodological research stems from ideas that come from very smart people who generate the data, and I love working. And there's a lot of them in Michigan, especially. I'm a rabid cricket fan, and I can be bribed with good coffee. My orthogonal existence is my family. I try to maintain a good work-life balance. So this picture right here is the four of us, my wife and my two boys, who are 10 and 12 now. And if you see that line is actually the prime meridian. So that was one of the high points of our trip last summer. So I'm gonna start at a very high level of medicine. I think it becomes more pertinent as you would be watching this through your Mid Students Day, especially in the current scenario. You know, where we are in terms of our healthcare and in terms of the whole healthcare enterprise. So there's a notion going around about this big P4 medicine, and how can we effectively, as quantitative scientists, leverage this. So the 4P stands for predictive, preventive, personalized, and then finally participatory. Okay, and this whole positioning of this, contextualizing this, and how do we as data scientists contribute to this, and holds a lot of promise. So with the promise comes opportunities, and I feel, and this pretty much encapsulates my phase when I started working in this domain. Around a decade or so ago, when somebody introduced me to this field, and it was a casual conversation, over a coffee, and said, we're collecting these levels of data at every level, and this is going to not only within diseases, but across diseases, and different types of genomics, transcriptomics, and going from genomics now, it's gone on to more levels in terms of the social media. So what's the challenge? So I think that's pertinent to us as quantitative scientists working in this field. So we're getting this voluminous of data, and one thing with this sort of data is it's coming at rapid levels, at different scales, from different platforms, at different modalities. And the key here is, and they're coming in at, even some of these are matched across for an individual patient. So the same patient or the same subject or the same experiment has been, same experimental unit. So the idea, the big picture idea here is, how do we extract biological or clinically implementable knowledge? Something we can understand about the disease domain, but also how do we actually eventually make an impact in terms of an actual endpoint in clinically translating this. So in my mind, and this one being an understatement, that integrating these data sets might just be one of the key quantitative challenges in driving modern science. So what is biostatistician's role in this play? And I was watching this movie a couple of weekends ago with my kids, and I felt like this access to this data set in this day and age, it's like a national treasure. And the clues are right there. And biostatisticians have a very unique training that when they come, and the training that you go through in most graduate programs, including at Michigan, is getting the know-how or getting the exposure to the whole spectrum of the data, the life of the life of the data. So that starts from how the data was collected and how do we actually model it, how do we quantify uncertainties, and how do we actually interpret them and provide those interpretations to a practitioner. In this big data world, it's literally panning for gold, and we have a very unique role to play in this with our expertise. So where does my group sit in all this? My group here in Michigan is called Base RX. So these 4 quadrants that you see on the screen, you can break it up by 4 quadrants. One is more on the discovery domain, and how do we discover biomarkers, and there are different methods of doing biomarker discovery, and then also looking at networks. And then the bottom right corner is how do we actually think about clinically translating this? And this has become a recent set of problems that has me consumed. How do we actually reach translational goals? And as a quantitative scientist, I feel methods development combined with effective, what is our product that we can eventually give for other researchers or the community to use, and that would be software and visualization tools. How effectively we can do that. The department does a great job on that respect. We have many extremely helpful resources and very knowledgeable people around who help out with this and developing these GitHub codes, shiny apps, and these databases, and how can we effectively leverage and do that, right? So I'm going to give you a few examples, some specific examples. I'll probably just give you a flavor of those as we move along. And like I said, I work mostly in cancer. So the cancer has a whole life cycle in terms of what are the precision medicine opportunities in cancer, or the precision medicine research in cancers, or more specifically precision oncology. So the circle here you see is sort of the life cycle of how research is done, how the data is collected, that it's analyzed, and sort of where we fit in. So my research and the students and my group that we work with is on data integration. So we have developed a few methods on how to integrate these different kinds of data sets, and how do we bring in some biological knowledge and structure in including this, and that includes both genomics and imaging. And as I mentioned earlier, one of my key pet topics is right now going into more personalized inference domain. So how do we do personalized inference now that we have this volume of data? It provides us a rich opportunity. So this concept of personalized networks in cancer that I've been working on, and a few of the students who are here working on. And finally, completing that cycle would be all the way from discovering what goes wrong at a very basic biological level, and how do we convert that to actually translation. So translational in this context would mean how do we effectively leverage drug targets, and what sort of therapies might be more effective, and learning that, mining that information. Right. So I'll give you a brief, like a high-level overview and a flavor of what I mean by personalized networks. So typically, when one starts doing modeling, one assumes there's a population. So this comes from actually a collaboration of mine with a multiple myeloma physician. Multiple myeloma is one of the blood cancers. It's one of the forms of cancer that forms in the bone marrow. And one thing, through our conversations, working with an oncologist, they wanted to understand, like, how does the genomics of a particular person change with the stage of the disease. So the different colors you see is the stage of the disease. As you go from green to more yellow to more pink/red is the more aggressive disease. So that's stage 1, 2, 3. And how do we build networks that might be representative of these different subgroups? So if you look at networks, these have different interaction patterns. So this could be networks among what are usually some genetic unit. So it could be genes, it could be other aspects also, or proteins, right? So the idea being that if you have two people who are sort of on the boundary, the yellow and the green boundary, would you expect a lot of changes in them? How do we effectively? So we came up with this concept of evolving networks, like as the disease evolves, looking at this as a continuum, and how do these networks change as the disease continuum. And it actually turns out it's a scientifically very interesting problem, but when you turn the crank, it is also a statistically very interesting problem, because you're trying to make an inference of a network from going from a population level to an individual level. So this was a work done by one of my former PhD students called Bayesian graphical regression, but how do we incorporate these type of biomarkers and study different type of networks across stages, right? And this is some more recent work done with a couple of students here at Michigan. This was a paper where we tried to do this, the same concept of personalized network modeling across both human tumors but also cell lines, which are models that are actually perturbed by drugs in laboratory settings. Right? And this was a recent paper done by Rupam that just got accepted for publication in Journal of Clinical Oncology, one of the top clinical oncology. And the basis of this was actually a method that can estimate this, but led to some really nice scientific insights. And this is another example of a project that another student of mine here in Michigan, Qingzi Liu, did, where we sort of came up with this network-based matching of patients and targeted therapies. So when we started understanding, like, how do we match the right patient to the right drug, we thought, why don't we use a very similar concept of like Facebook? You know, if you're a friend of a friend, and then if if two people have a large set of common friends, then naturally there is a recommendation that you should be linked to each other in some sense. So it's sort of a similar idea that we have built into matching these targeted therapies. Is these targeted therapies are studied on cell lines, and they have patient levels. So if you can build a network across these different sort of what are called model systems, then you can figure out that missing link that shown in blue here between a particular patient and a particular drug or a set of drugs. So why this would be useful? It would be useful to rank drugs, or rank drugs for a particular cancer, but also rank drugs across cancers, or rank multiple drugs that might be given to a patient. And this is all data-driven, evidence-based, mining all the data that we get. And as I said, one of the key projects here is developing how effectively these tools can reach the community. So part of that is developing apps. So that's an app that Qingzi developed, and it's a really cool visualization of inputting a particular patient shown in the brown bar, the brown vertical bar, and what are they matched to the top drugs. Right. So this is a very exciting line of work, and the future, in my opinion, is very bright, and it involves the 3 I's here: the integration, innovation, and how do we actually do implementation. And I would like to acknowledge a lot of people here. It took a village to get what I'm showing as a small snapshot of this work. So as you see here, if you had been here, this is the front end of the School of Public Health. And that yellow dot that you see, that's my office right there. And if you see it, I'm sort of on the bridge between biostatistics, which is on the right-hand side. We are on the 4th floor there. And sort of when you move more towards the left is the whole medical school enterprise here. And metaphorically or literally, I love being on the bridge because I'm a bridge statistician, and I work with a lot of clinical basic science and translational investigators, as well as some stellar students and collaborators in the Department of Biostatistics. And hope you found this informative, and then I'm hoping that you would consider us and be a part of our team sometime in the future. Thank you, and I wish you all the best, and I'll be happy to please get in touch with me. My email is veerab@umich.edu, or on my website there's a contact, and I would love to hear from you if you have any questions or clarification. Thank you, bye-bye, enjoy, and welcome to Michigan. Go Blue.